a test tube, right? Do you think we can grow a zygote in a test tube?
Not grow the baby in test tube. We make these things in test tube.
So it's called a test tube baby, which is kind of a misnomer in the field. People think that. So test tubes sometimes have nothing to do with it, to be very honest. Okay. So what happens is, you're right. Rest part, you're correct that we take gametes from the male and the female, correct? And all we do is we allow it to fertilize in vitro. What does in vitro means? The word in vitro. There are three words: in vitro, in vivo, in silico. So what does in vitro means? Any idea?
No, sir.
So you can write down these three things. It might be of might be useful for you to know. So in vitro means, vitro simply means vessel or apparatus or glass vessels. Okay. So in vitro means in anything outside the body, inside a glass apparatus. Is it clear?
Yes, sir.
That's in vitro. In vivo means inside the body. Okay. Inside the system, living system. That's in vivo. Are you writing these things down?
Yes, sir.
Yeah. And in silico means digitally, like through computers and softwares. In silico means inside the computer or software. An example of all I will give to explain is, in vitro is like any chemical reaction that you do in your chemistry lab, you are doing it in vitro. Okay. Because you're doing it in glass vessels, test tube, outside the living systems. For example, if you take any sugar or carbohydrate in a test tube and break it down with some catalysts, you do a reaction inside a test tube. That's called an in vitro reaction. If the same reaction where the glucose is broken down by enzymes in your body, inside our cells, inside every living cell, then it is called in vivo. Okay. And if you are simulating a software where you are doing something, for example, you are again simulating this reaction and studying this reaction through softwares in computers by artificial intelligence or something, then it's called in silico. Make sense, Alan?
Yes, sir.
Perfect. So that's where genetic engineering is when we go in vivo and we tweak the genetics of a living system, bacteria, cell, human cell, or any gene of human or bacteria. And bioprocess engineering is everything outside of living system. For example, in IVF, we don't tweak the genetics, do we? No, right? We just take a sperm and an ovum. Do we make any changes in their genome?
No, sir.
No. And for that matter, it is not allowed. It is not allowed to make changes in the genome of humans. Genome editing in humans, it's not allowed. Okay. So what do we do? Why we call it a biotechnological process is that everything apart from that comes under bioprocessing, which we do. We take the gametes with some technical aid, with some apparatus instruments which we have created, and they should be kept in very sterile environment. And when we are allowing them to fertilize, that fertilization also should happen in a sterile free, without contamination. No bacterial, no viral contamination should be there. So all these come under bioprocess engineering. So these are the two core techniques by which biotechnological processes work. Is it clear, Alan?
Yes, sir.
Yeah. Okay. Then from there we went on to talk about them in detail. So what did we talk about in gene cloning? Can you tell me what do we mean by gene cloning? What was gene cloning?
So we talked about different types of this side, such as origin of application or the ORI, and then we talked about how plasmid vector or a bacteria is autosomally replicating circular DNA, it's used for changes in the host that we want to introduce it to. So a plasmid vector carries the mutation that we want to express in the host, and it breaks the DNA cell wall by using different methods such as the thin gold foil method or microinjection. And then we talked about how there are different types of gene cloning, such as there are different types of cloning such as PCR and so on.
Mhm. Right. So in general, you're correct. So when we are cloning a gene, simply, well, what you could have said, what I wanted you to say, cloning simply means what? You are correct. You explained the whole process correctly
Making multiple
without.
copies.
Yes, perfect. Cloning simply means making. Remember, we also studied clones when we were talking about asexual reproduction. Do you remember that? Bacteria, when bacteria divides by fission, technically all bacteria are clone of each other, right?
Yes, sir.
The daughter bacteria is a clone of the mother bacteria, right? Bacteria. Yes, so cloning simply means making multiple identical copies of the gene sized pieces of DNA. Okay, so we are interested in one gene. It could be any gene. It could be the gene for the ADA enzyme. It could be the gene for insulin. In case of making insulin for diabetic patients, it could be a gene for any other enzyme that we want to cure a disease or make a medicine or simply to make proteins. Okay, so that's gene cloning. Now for that we need a, so to clone any gene we need to take that gene out, right? So now let's quickly go to the process of cloning of the gene of interest and constructing an artificial recombinant DNA molecule. These two topics are the most important topics from this chapter, and basically this is the foundation on which this chapter is based on. So we talked about plasmids. The process you have already explained in good detail, but what is the role of endonuclease? I also gave this as a question in one of the exams, right? So what are endonuclease and exonuclease? What in general are restriction enzymes? Would you like to quickly go over it, Alan?
Yes, sir. So endonuclease is the nucleic acid that digests the enzyme inside the nucleus, and exonuclease is outside it. As for restriction enzyme, it cuts the
Not
nu-
inside or outside the nucleus. Nucleus simply means the one
the
that
cell,
breaks nucleic acid. Yeah, but inside or outside what?
the cell wall.
No,
Oh,
no,
so
no, no.
because I wasn't there in this
So
chapter. I joined.
look at this figure. What I'm doing here. What are we doing here?
So over here we break the DNA strands.
Uh-huh. So I have labeled
Okay,
exo.
okay. So an endonuclease cuts it from the center, while exonuclease cuts it from the ends of the DNA strands.
Perfect. So the word endo means inside, towards the inside. So this region is inside of the nucleic acid, right? This is the
Yes,
outside,
sir.
right? And nucleus simply means nucleic acid digesting enzyme.
Okay.
So endonuclease is a restriction enzyme that cuts any nucleic acid, for example DNA, anywhere in the middle at a specific site, at a specific sequence. And a exonuclease is a restriction enzyme that does not cut, but it removes or it chews the DNA from the ends. Okay, it starts degrading the DNA from the ends, but it cannot cut in between somewhere. That's why it's called exo, and the other one is called endo. Is it clear?
Yes, sir.
Yeah.
So a restriction enzyme cuts the DNA, and it
Yeah.
when it cuts it, it forms sticky ends or complementary strands of the hydrogen bonds, and
Yes.
then we add that two complementary strands of DNA to form the new sequence.
Yes. Perfect. Now you're right. Good. And how do we name them? What is this? We also studied their nomenclature.
Yes, sir. So the first word will come from the for eco R1. The first word from the prokaryote, if I'm correct. E. coli, and then R will be the strain of it, and then 1 will be the order of discovery of isolation.
Correct. So the first 3 alphabets, you mean to say, let me be again clear, is
Yeah, so the first 3, yeah.
first 3
Yeah.
alphabets. Among the first 3, the first is the genus name of the, so you can say, you can write if you have not written in eco R1 or in any restriction endonuclease name, the first 3 alphabets stands for the source organism. Okay, scientific name of the source organism. Okay, so E is the genus of the source organism, which is Escherichia, and CO is the species of the source organism, which is coli. Make sense?
Yes, sir.
Then R, as you correctly pointed out, is the strain, in this case RY13, and 1 is the order of isolation or the order of discovery. Okay. Very well. Same is for hint. So this part is done. And yes, one question which I also gave, and it often comes in boards, is if they ask you why restriction endonucleases only recognize palindromic sequences, what will you write?
So for a palindrome is a sequence is at the same backwards and forwards.
Yeah,
So
first,
for
what is a palindrome? A palindrome, yes, you're right.
a palindromic sequence is a DNA that is read both the same backwards and forwards if it's the same
Perfect.
sequence.
Yes.
And then for restriction enzyme, it cuts the DNA in complementary hydrogen bonding. So for a new DNA to be formed, we need the complementary things with the complementary base strands. So a palindrome, since it's the same, when two palindromes join together, they'll be complementing each other from the opposite strands, and they form a new dsRNA.
They form a new.
A new a dsRNA, double-stranded
Hmm?
RNA. No, wait, was
No,
it
no, but
D?
no, but yeah, you're right. But that is not in this context. In this context, why the restriction endonuclease recognize and only cut at palindromes is because, as you said correctly, palindromes read the same in both, like in both the from right to left and left to right, both orientations. And these restrictions endonucleases cut not at the center of the sequence they recognize. They cut slightly offset towards the left or towards the right. But the benefit with palindromes is if you cut on one strand, one base towards the right, the same base will be there, one base towards the left on the next strand, right? Do you understand?
Yes, sir.
Yeah. And this, what it does it, recognizing and cutting at palindromic sequences leaves sticky ends. Do you remember sticky ends? The importance of sticky ends?
Yes, sir. The unpaired hydrogen bonds left over from cutting of the hydrogen bond.
Yeah,
But the
correct.
so see, since they are unpaired, they'll come to join together and form a complete strand.
Yeah, they can be easily joined by the enzyme ligase, right? Yeah, correct. Yeah, perfect. So they leave sticky ends, these restriction enzymes, when they cut at palindromes. So remember that. So the sticky end is very important. Okay. Is that okay?
Uh,
Okay, cool. Very good. So this concept is done, and um, this was the whole process that we discussed. Let's go over it once again. I will go over it and you tell me at every step if you have any doubt. Okay.
okay, sir.
Okay. So this is the foreign DNA, correct? So in that foreign DNA, we have a region of interest. So what is this region of interest?
That's a place where we want to introduce changes to. It has a few sites that are easy to identify.
Yeah, so basically this is the region of the DNA which we want to clone, remember? Yeah, remember we want to, for example, this could be the gene of insulin or anything, right? So always remember these terms are common and they will be repeating over and over again in this chapter. So this region of interest, we have to use the same restriction enzyme to cut our foreign DNA and to also cut our plasmid. This is a line you should always remember. Okay, Alan?
Yes,
And if
sir.
they ask you, so this is a legit question that has come in exams where they ask, why is it important to use the same restriction endonuclease or same restriction enzyme to cut the gene of interest and also to cut the plasmid vector? So this is important so that both plasmid vector and the gene of interest have the same sticky ends, because if they will have the same sticky ends, they will ligate better. Make sense?
Yes, sir.
Yeah. Okay, perfect. And what happens after that? We use ligase enzyme, and we through ligation we put our recombinant DNA, which is shown here in the black. Can you see the figure? This figure.
Yes, sir.
So what's shown in the black is our gene of interest or our recombinant DNA, and sorry, is our gene of interest, and we have put it into the plasmid, which is red, and this makes our recombinant DNA. So this whole DNA containing plasmid and our gene of interest is called recombinant DNA, because we have created it through recombining our gene of interest with the plasmid. You understand why it is called recombinant DNA? It is a product of recombination, right? Is that clear, Alan?
Yes, sir.
Yeah. Okay, perfect. So what after that? The recombinant DNA is in our with us. Now what do we want to do? What does the next step shows here?
We are interested into the bacteria or E. coli and how it transforms, and then we clone it to produce multiple copies of the desired DNA.
Yes, perfect. But just before that, how do we put that in the bacteria? Why will the bacteria take our DNA?
So if it's incompetent, we make it competent, or else we just take it.
Yeah. So we make our bacterial cell competent. We call
So
that as.
So we make it
Yeah.
competent by like a few methods there, such as microinjection, and then there's also there's there's a gold foil method in high velocity.
Not gold foil. It's
Not
called
gold
a
foil.
gene gun.
Gene gun, right?
Where gold and tungsten nanoparticles are used.
Yes, sir. So
You coat the nanoparticle.
high speed temperature in the cell wall.
Yes, perfect, correct. So remember then, which one is used for which, okay? Just keep that in mind. Yes,
so
and
what,
then.
So what do you mean?
So remember I asked a question in the exam that which method you will use to make a bacterial cell competent, a plant cell competent, and an animal cell competent.
Okay, like the different types of yes.
Yes. So do you remember, for a bacterial cell we will use chemical method, where we use calcium or any divalent cation. Okay.
Okay.
Calcium. It's called calcium and heat shock method. Remember that? Calcium and heat shock method.
Yes, sir. Yeah. If at first we heat it and then cool it and then heat it again.
So we first cool it on ice, then we heat it for a brief time at 42 degrees Celsius, and then we again put it on ice. And before doing that, we treated it with calcium chloride or magnesium chloride. Okay.
Okay, sir.
That is for bacteria. It's called chemical method or heat shock method. Second, for plants, we use gene gun. It's called biolistics, right? Gene gun means what you said. Coat a gold or a tungsten nanoparticle with your DNA and bombard it or at very high velocity. Just shoot the plant cells with that. So some might enter inside the plant cell. For animals, we use, the one is left. Which one do we use? Microinjection.
Microinjection.
Okay, because in animals it's easier because they don't have a cell wall.
Cell wall.
They just have a, yes, perfect. Very good. And then you put it in the bacteria. So here it is bacteria, and then the bacteria will divide. And when it will divide, it will also replicate our recombinant DNA, and that will lead to cloning of our gene. So this process is called gene cloning. Simple. Okay, makes sense?
Yes, sir.
Any doubts in this?
For the bacteria, if it's incompetent, we use the first method, heat shock, right?
Yeah, the chemical method along with the heat shock. So
Okay.
first treat the bacterial cell with divalent cation.
Cation
It can,
or calcium.
yeah,
Yes.
it can be calcium chloride or it can be magnesium chloride.
Chloride.
And then you do the heat shock with the plasmid present. So through the heat shock method, the plasmid will enter into the bacterial cell.
Okay, sir.
Okay. Any part which is confusing?
No, sir.
No. Okay, perfect. Okay, then we'll move forward, and we talked about gel electrophoresis. Yes, another method. Anything problematic here?
No, nothing, sir, over here.
No? Okay, cool. So just remember that in gel electrophoresis, the DNA moves because DNA is what charge? What is the charge on DNA?
It's positively charged.
DNA is negatively charged. DNA is
yes,
always
yes, sir,
negatively charged. Just
yes, sir,
remember
because
that.
of the histone,
Because of the,
right? Histone.
no, because of the phosphate.
Okay.
Yeah, DNA has a phosphate in it, right? Histone is the protein. Histone is positively charged. You're
Okay.
right. So that's why DNA can bind around histone and become compact and make a chromosome. Remember I told you that.
Okay. Yes, sir. Yes.
Yeah.
Yes, sir. Yes, sir.
So DNA itself is negatively charged because of phosphate molecule in it. Histone is a positively charged protein. Okay. And that's why DNA can bind around histone. And for the same reason, when we do electrophoresis, we just take DNA, not the phosphates. Okay. So DNA will run from the negative terminal, which I have shown here. Can you see? Towards the positive terminal because itself is negative charged. Okay. So in a gel electrophoresis, DNA moves from negative terminal to positive terminal. And the farthest a DNA is, the smallest will be its size on that gel. So smaller DNA particles will move the farthest. Obviously, right? Because they are small, they can easily cross the pore with the same amount of voltage. Okay, make sense?
Yes, sir.
Okay, very, very good. And no doubts, right? Okay, and yes. So this is the answer for the question. Can you read the question and the answer and tell me if you understand it properly? Can you read the question in the blue and the answer in black?
Out loud?
Yes, yes.
Okay, so
just,
what?
Yeah. You can just read it for yourself also, not a problem.
Okay,
Yeah.
sir.
Just let me know if this is clear, all the three things. If you have any doubt in any of the three things, let me know.
No, sir, it's fine.
It's clear?
Yes, sir.
Very well. Okay. Then we just simply figured out how to. So these are the steps after studying everything. What we have to do again, we have to isolate the genetic material, and everything is clear because this was also your practical, right? In
Yes,
your
sir.
schools, you all have
Yes, sir.
isolated
Yes, sir.
DNA. So from which source you have isolated DNA?
Sir, strawberry.
Strawberry, right? I think we discussed that. Strawberry, you can isolate DNA from strawberry. Perfect. But this is just going into, so we cut, first we isolate, break open the membrane. Okay, we use these enzymes. When we are using a plant cell, we use cellulase, chitinase, or lysozyme. But in your case, you just use detergent, right?
Yes,
Detergent is in general a agent that breaks the cell wall.
that's the cell wall.
Yeah.
And
Perfect.
then
And then yeah,
and then we add chilled ethanol for precipitating the DNA.
yes, perfect. And it should be chilled ethanol, carefully
Allow size
poured
of test
from
tube.
the
Size
sides
of
of
the
the test
test tube.
tube. Right. Yeah. Because otherwise, once it get mixed with the water, you won't be able to
We the
precipitate out
precipitate.
DNA.
Yes, sir.
Yeah. Yeah. Perfect. And then what we do once we have the DNA in of isolated from the source, we don't want the whole DNA, right? The whole DNA is of no use to us. We only want a gene from that DNA, which is important, right? So
Yes,
we
sir.
cut the DNA at specific site wherever we want, whatever part we want, through restriction enzymes, right? We use the same restriction enzyme to cut the DNA and the vector both, as I told you before. Then third thing, sometimes before we put it in the bacteria or a plant or animal cell, wherever we want it, we amplify it beforehand. And for that amplification, we use a technique called PCR. Correct? Can you tell me what is PCR? What does PCR stands for?
Yes, sir. PCR stands for polymerase chain reaction and it consists of three steps, which is denaturation, annealing, and extension. So
Mhm,
for these things, we use several enzymes, and then there are DNA primers. So the enzyme we use is Stack polymerase. We use Stack polymerase because it is found in hot temperatures such as heat vents and volcanoes. So it's able to withstand a
perfect.
hot temperature.
Mhm,
And then for first we denature it in heat, and then we we anneal the oligonucleotides or the DNA primers with the
mhm,
Taq polymerase, and then we extend
mhm,
the DNA strands. And
mhm.
after we get the copies of it, we make several copies of this by repeating PCR over and over until we get the desired number of copies.
Perfect. So that's perfect. So three steps are very beautifully explained. Very good. I think PCR is clear to you. And then fourth step is, after we have enough amplified copies, we insert it into the host cell, and for that um, we make the bacterial cell competent or any cell competent. And we just discuss the methods which we use. And also we use a selectable marker. Can you tell me what is a selectable marker? Because that could be a 2-marker question.
Yes, sir. So a
What is
selectable marker is mainly used for usually recognizing the chlorine vector. So for example, in PBR 322, we have the selectable marker
it is to recognize which cells have
taken
successfully taken
successfully
our
taken up the.
plasmid vector.
Yes, sir. So a
Yeah.
selectable marker in PBR 320 would be the ampersand resistance or the tetracycline resistance.
Mhm.
So, uh, so if we, for example, if we take up an
So basically, how will you know that your plasmid is inside the bacterial cell? If your plasmid, apart from your gene of interest, has something else, for example, another gene that gives the bacteria that has taken the plasmid another advantage. For example, if your plasmid has an antibiotic resistance gene, then apart from um, your gene, it will also be producing the antibiotic resistant molecule protein in its body. So your cells which have taken your plasmid will survive in antibiotics, but others will not, right? So that way, antibiotic resistance gene becomes a selectable marker gene, correct?
yes, sir.
Do you understand that?
Yes, sir.
Yeah. Pakka. And then the final product is obtaining the gene product, a foreign gene product, where we allow them to grow in large bioreactors and control their growth conditions, etc., and obtain the protein. And then comes the downstreaming process, where I told you that there are two things that come in the downstreaming process. One is called filtration and like purification and isolation, right?
Yes, sir.
Okay, very well. So this is also this in this chapter you have no doubt, right? No doubt as such.
No, sir.
Perfect. So let me stop sharing this and I'll give you. So can we take a quick 2 minutes break? I'll just get some water for me and then we'll start
Yes,
with
sir.
some questions. Yeah,
Yes, sir.
Helen? Questions from this chapter we'll take, okay?
Sure,
Yeah,
sir.
so I'll resume in a couple of minutes. You can also utilize this time or just look through your notes if you have any doubts. Okay, I'll be back. Okay, are you there, Helen?
Yes, sir.
Yep. So shall we begin with some questions?
So I'll just revise for like a few moments and let you know.
Yeah. Okay. So let me give you 5 minutes. How about that? We'll start exactly after 5 minutes,
Yes,
and we
sir.
can go on for half an hour. Huh?
Yes, sir.
Okay. Perfect. So yeah, time starts now. Okay. Take your time.
Yes, I'm done revising.
Okay, you're done, Alan?
Yes, sir.
Okay, so let's start with some questions. Okay, let me share the screen. Just 1 second. So again, we'll follow the same thing. I'll go for some complicated, conceptually rich questions. We'll not go for. We'll do some just factual, important factual questions. Okay. Okay. Can you see the screen that I am sharing, Alan?
No, sir. Yeah, yes, I can see now.
You can see, right? Okay, perfect. So cool. Let me tell you which one. Okay, perfect. So let's start with some simple one. So because it's you and me, so you can just read and you can tell me the answer whenever you feel. So try to be fast, which you are in most of the time, but also try to be correct. Okay, so this time we'll just get one go. Okay, so whatever will be your answer, I'll just tell it's right or wrong, and then we'll discuss if you need discussion. You
Yes,
can
sir.
also choose not to select an option and say that you need to discuss a little bit. Then what we'll do, we'll discuss without me telling you the answer, and you can tell me the answer. Okay, so let's start with question number 8. Can you let me just make it a little bigger? Yeah, I think you can see question 8.
Yes, sir. That'll be option C.
Yes, it's so one of the key factors which makes a plasmid vector in genetic engineering is its ability to carry a foreign gene, right? So it's so key factors, when we talk about key factors, okay,
Most important factor of all the four.
yes, so this is also true that it's resistance to antibiotics. Okay, it's not resistant to an restriction enzymes. That's wrong.
Yes,
Its
sir. Both.
ability to cause infection in the host can also be true sometimes, but
Yes, sir. If we,
C is the most important one, right?
yes, sir.
Yeah, very good. Great. So next I've chosen is question 11. Read and let me know. If you will read, it will also be good for me. Like if you read it loud, because we both are reading otherwise.
Sir, 11, right?
Yeah.
Okay, so which of the following contains the key tools for recombinant DNA technology? Restriction
Yeah. That's it.
endonucleases.
That's it. So
Okay.
don't read the options. Just read the question so that I also
Okay, sir.
hear the question. That's it. Yeah. Thank you. Yes. What do you think?
So 1 and 2.
Remember again, it asks. No, the
Yes, sir.
options are below.
Oh, okay. So it'll be yes, I'll be
A,
B
B,
then.
C, and D.
So it'll be B 1 and 2.
Option B, 1 and 2.
Yes, yes, sir.
Yes, correct. Perfect. So these are the two key tools, right? Why
Yes, sir.
3 and 4 are not the answer is because 3
So 3
contains
will
this
be,
tag polymerase.
yes, that's used mainly for PCR.
Yes, right.
And 4 has
And 4
bioreactors, which also like for
bioreactors. Correct. So it's not important for recombinant DNA technology, right?
yes, sir.
Yeah. It is for making
For
that gene product, obtaining the gene product.
yes, sir.
Perfect. Okay. So see, a question like this has also come just to show you that the letter R in eco RY R is derived
That'll be
from.
option B.
Yes. So option B, the name of the strain RY13, right? I want you to. Can you quickly tell me which of the following is a palindromic sequence in the following?
So option B.
The lower and the bottom option? B, right, perfect. You're fast at it.
Yes, that's
But
option B,
this is
right?
simple. Yeah, it's option B, correct. Okay, read question 18. Question 18.
Okay, so read the given statement inside the correct option. Restriction endonuclease enzymes recognize a specific palindromic nucleotide sequence in the DNA. Restriction endonuclease enzymes are called as molecular scissors or biological scissors. So it'll be option A, both are correct.
Yes, both are correct, perfect.
I was
Okay.
looking for the A and R were like now the correct explanation, but it wasn't there in this one.
Sorry, you're looking for?
I was looking for like the common type. I was like it'll be like A and R are true, but the correct explanation, not the correct explanation.
Yes. So sometimes they ask is that they are called assertion reason questions.
Yes, so
So
for
what they
this one
say
will
is,
be not the correct explanation, right?
yes, so this one is just about independent statements. So the options you will see, you see that the options are either both are correct, both are incorrect, one
But
is
then
correct, the other
it
is
was
incorrect.
asked like the first one being assertion and second one being reason. Then will be
So,
option B, which is like assertion is true. I mean both are true, but ours are the correct reason.
but yeah, but statement is not the correct. Statement 2 is not the correct reason for statement 1, right?
Yeah. Yes, sir.
That way you're correct. You might also, we'll also do some assertion reason questions if time permits, but let's first go through this one. Okay, question 26. What do you think? Simple question, but concept based. You read question 26?
Class one vector is diagonal with equal R1 and single side, then so our plasmid vector is circular, so I think it'll be two sticky ends.
So you mean, so which option?
Option B.
B, two sticky ends will be produced.
Yes, sir.
Correct. Correct. So at a single side, remember where it cuts? It cuts at a palindromic sequence and it cuts little away from the center. Okay. So when you cut at a palindromic sequence and you cut a little away from center, you leave two sticky ends on each side, right?
Yes, sir.
Perfect. Okay. 27 in along the same line. So I'm just 27. Read question 27.
How many fragments will be generated if you digest a linear DNA molecule with a restriction enzyme having 4 recognition sequence on the DNA?
Yeah.
So I think it's option B6.
Think again. See, it's a linear DNA molecule.
Wait,
That's the point.
it's 5, right? Then option C.
Yes, it's 5. If it's a circular DNA molecule,
Then it will be 6.
yes, right? Do you understand the
Yes.
difference now?
Yes, I was like drawing the thing and checking
Yeah.
it out.
So it's very simple a question, but if you good that you are drawing. If you don't draw it, might be you just end up choosing a wrong answer. Okay.
Yes,
Perfect.
sir.
Good. Very good. So the next one that I've chosen is okay. Question number 33.
Having become an expert on gel electrophoresis, you are asked to examine a gel for a colleague. Where would you find the smallest segment of DNA? So it's either option A or C because smallest will be the most digested, but I'm not exactly sure which is if it's positive or negative actually.
Yeah. So think about it. If it is going far from where it began, so it is moving to. It should move towards the positive, right?
There'll be
Because DNA itself is negative charged, so it can only move towards the positive terminal.
positive. Okay, yes, so that'll be option A.
Yes, it's option A. Okay, the farthest DNA can go towards the positive terminal because that is on the farther side. if what happens if you reverse the terminals, if you put the gel in a reverse position, the DNA will come out of the gel and will just fall into, it will not move towards the negative, towards
Okay,
the
because
farther side.
it'll become repulsive, right?
Yeah, right. So it can only go towards the positive terminal, right?
Yes, sir.
Yeah. So it's option A. Perfect. Okay. And this is simple. I think you'll do it. Yes. So can you see question number 38? You have to little just analyze it. Just it says that which of the following statements are correct. Then there are 5 statements and multiple are correct. So you have to tell option A, B, C, or D. So read and take your time and give me the answer. Okay. Question 38.
Yes,
Are you
sir.
doing it, Ellen? Yeah. Okay.
Yes, sir. So I'll be option B, 1, 2, 3, and 4.
Option B. Yes. Perfect. It's option B, 1, 2, 3, and 4. Just 5 is incorrect because it says DNA is a positively charged molecule,
Yes, sir.
and which we discussed in today's class only,
Yes,
right?
yes, sir.
So
Yes,
pay
sir.
this in
DNA is
mind.
negative.
Otherwise, yeah, otherwise you would have chosen option D,
Yes, sir.
right? Yeah. So DNA is negatively charged. Okay, histones are positively charged. Perfect. Very good, Ellen. You're very good with that. You're picking up fast. So let's do one more along the line. Question number 40. Again, read and tell me.
So I'll be option C, 3, and 4.
It will be option C. 3. 3
C.
says yes. 3 is right. 4 is also correct. Yes, perfect. Now, would you also tell me why 1, 2, and 5 are incorrect?
Yes, sir. So the first one will be correct if we change it the last line to same the same enzyme.
Yes,
So
right.
first is wrong, and second will be correct if it's an endonuclease, but exonuclease is not the correct answer.
Yes, correct.
And for
Fifth,
fifth one, if there are more than one recurrent sequence, for gene cloning, we'll obtain many multiple fragments, which will complicate the process. So we usually take like 1 or 2 recurrent sequence.
yes, right. Perfect. Very good. So again, question 45. So let's answer it both ways. One, what the answer question is asking, and the second is assertion and reason also, right?
Okay, sir.
Okay. Would you like to read the two statements?
Statement 1, the cloning vector is required to have very few, preferably single recurrent sequence for the commonly used restriction enzyme. Presence of more than one recurrent sequence within a cloning vector will generate several fragments, which complicate the process of DNA cloning. So it'll be option A, both are correct. And for assertion reason will be again option A. Assertion and reason are true, and reason is the correct explanation for A.
Yes, right. In this case, reason is the correct reason for the assertion.
Yes, sir.
Right. Very well. Good. So we will move a little towards. Yes. Question 51.
Using recombinant DNA technology, genes from a donor cells can be inserted in a bacterium for DNA replication. and photosynthesis. The kind of cells that have been used as gene donors in technology are
Remember, they are inserted it into a bacterium. They are inserting it into a bacterium and asking what can donate for that purpose.
so I said option C.
Eukaryotic? No, it is option D. Any of these. So remember, any gene from any organism you can use and put it into the bacterial cell. Even the genes that are not bacterial, which are not present in the bacterial genome itself. So it can be from bacteria, it can be from yeast or bacteria, it can be from eukaryotic cells, it can be from any of these. Right? So basically it can be of any of these. Not just we in NCRT have always talked about using genes from humans. Does not mean we can only do it with genes from humans. Okay, so that is a difference between how we learn and what we should know. So even if you want to take a gene from a fungi, a yeast or a fungi, and clone it into a bacteria and then use it for human purpose, that can also be done. The answer will be D. Okay, any of these. Is it clear?
So yes, sir.
We'll keep it in mind, right?
Yes, sir.
So yes, one question on insertional inactivation. Again, question 56. Read it. I gave you this as a homework to read. So let's see. If you need a discussion on this, you can let me know. Question 56.
Statement 1. In insertional inactivation, blue color produced by the bacterial colonies indicate the plasmid does not have an insert in the bacterial genome. Statement 2. Presence of insert results in insertional inactivation of B characterase enzyme and the colonies do not produce any color. So here, pretty sure both are correct.
Yes, you're right. Both the statements are correct. Okay.
And the reason is also statement 2 would also be the reason for statement 1.
No. So the reason statement 2 is not the correct reason for the statement 1. Here both the statements are telling you the opposite things of what is happening. So if during insertional inactivation, if you get blue color from the bacterial colonies, it means that the plasmid has not entered into your bacteria cells. Okay, so the plasmid is not present in the bacterial genome. Is it clear? Second statement says that presence of insert will into the insertional inactivation of beta galactosidase enzyme, colonies do not produce a color. Correct. But two, the correct reason for one is that when there is no insertional inactivation, the gene of beta galactosidase enzyme is active. It produces beta galactosidase, and that reacts with the substrate present in the media, which gives a blue color. The blue color is only produced when beta galactosidase enzyme is active. So that should be the correct reason. Statement 2 does not says that. Do you understand?
Yes, sir.
Statement 1 tells you what happens if insert is not present. Statement 2 tells you what happens when insert is present. That's why statement 2 is not the correct reason for statement 1. The correct reason for statement 1 is that when insert is not present, the enzyme is active, it reacts with something in the media and gives a blue color. Okay. Does that make sense? Is it clear?
Yes, sir.
Yeah. Okay. Perfect. So I'll move from here. I have one more part. Processes. Yes, this one. Question number 69. So again, there are 5 steps given. There are different steps of recombinant. They are given randomly. You have to tell the correct sequence. That's all. So read and take your time and let me know. Which of the ABCD option is correct? What is the correct sequence?
So it'll be option C.
Option C, yes, correct, perfect. So all you have to do is to figure out what is the first and the last.
First one and the first. Okay, I saw the first and the second one.
Yeah, so the first, when you
So
read all the statement, so you have to first read all the statement, then only you will get to know
the first one.
because then only you will get to know where is the process starting and where is the process ending. So statement 3 is where the process is ending in this case, multiplication, expression of the introduced gene in the host. None of the statements talk about downstreaming process, right?
Yes, sir.
If any statement would be talking about downstreaming process, that would be the last in the sequence. So here, according to these 5, third is the last in the sequence, and which happened to be only an option in option C, right? You see, third is the last. Here, fifth, fourth, and second. So that way it becomes more easier. That's how you do these questions faster. Okay,
Yes, sir.
remember this trick. Okay. Okay, so another conceptual question. Yes, question 78. What are primers?
So the option A.
Yes, perfect. It's option A. Chemically synthesized oligonucleotides that are complementary to the region of DNA. Okay, what's wrong in B, C, and D? B says not complementary, so it's wrong. If it is not complementary, it will not bind. If it will not bind, it cannot start the process of PCR. Simple.
PCR.
C, it says autonomously replicating. That's not possible. Oh,
Yes,
sorry,
sir.
that's possible. But when it is a autonomously replicating circular DNA, it's not called primer, it's called plastid.
Plasmid.
Sorry, plasmid.
Plasmid. Yes, sir.
Sorry. So C is wrong because it's the definition for plasmid. D, specific sequences present on recombinant DNA. No, that's not primers. Okay, those are
Yes,
called
sir.
either region of interests or selectable marker genes.
I don't know. Okay.
Perfect. Very good. So, and then question 80. Read that. Read that for me. What does it?
Question 80.
Yes.
So which of the following statements are correct for enzyme Taq polymerase?
Yeah.
It remains active during the high temperature induced during denaturation of dsRNA. It requires primers for carrying out the process of polymerization. It synthesizes the RNA region between the primers using DNTPs and Mg2+.
Yeah. Which do you think is correct here?
So option A, 1 and 2.
Option A, 1 and 2. Let me read 1 and 2. 1 is correct. Yes, correct. What is problem with 3? Why do you think it's incorrect?
So option 3, I just Mg2+ is just using for the competent, making the host cell competent. I never saw this line during the PCR process steps.
No. Option C, option 3 also would have been correct option if, see, they have written Taq polymerase only synthesize DNA, not RNA. But they have written it synthesizes the RNA region.
Oh, okay.
If you change the word RNA to DNA, option 3 is correct. It does synthesize the DNA region between the primers using DNTPs and Mg2+. Okay. The only word that's making it wrong is the word RNA there. Is that clear, Alan?
Yes, sir. Yes, sir.
Perfect. So let me also spend some few minutes on some hot questions like high order thinking skills. So these are the questions that I have found to be very, very interesting questions. Okay. For example, there was one. Yes, this one. Question 3. This is one good question I found. Can you see question 3? And it's
So I'll just see the question first and then go for answers.
can you see question 3 here
Yes, sir.
completely? Can you also read it, or is it difficult for you to read?
Once I can read it.
Okay. So would you like to read and analyze this? I'm giving you 2 minutes for this question, not one.
Okay,
Two
sir.
or okay.
So I'll be option C, colony 4.
It will be option
C.
C. Yes, it will be option C, colony 4. So I think you understood the whole thing. So the reason you chose colony 4 is because that is the only colony which is growing on agar, but not growing on tetracycline, right?
Yes, sir.
So because the recombinant DNA is introduced in the tetracycline gene resistance region. So now that becomes non-functional. Okay, let me ask you one question. What else they could have done to show, like give also a proof that you just did not miss colony 4 because of any issue? They could also just make one more plate of ampicillin, right? If I
Yes,
make a
sir.
plate of ampicillin, what all colonies will I see growing on ampicillin?
Only 4?
No, all of it,
Okay.
right? Because all will have ampicillin intact,
Oh,
right?
okay. Yes, yes, sir.
Yeah. So actually plate M is basically same as of ampicillin plate. Okay. That is one trick thing you should just remember. Okay. So I think concepts are quite clear for this